Hello everyone, welcome to week 15, lecture 2. In this lecture, we'll discuss about cloning and biotechnology, exploring the science and its application. Let's first look at some definitions. Cloning is the process of creating identical copies of an organism, cell, or DNA molecule. Biotechnology is the use of living organisms, cells, and biological systems to develop useful products and technologies. The applications of science in the form of cloning has been used for quite some while now. The first experiment on cloning with frogs was performed in 1950s, and Dolly sheep was created in 1996 through the process of cloning. Cloning is mainly of three kinds. Reproductive cloning is when we create the whole organism that is identical to the donor. For example, Dolly sheep, which was created from another sheep. Therapeutic cloning is when we clone cells, tissues, and organs for the medicinal purposes. For example, cloning stem cells to regenerate new organs or even blood for some patients. Gene cloning is copying specific segments of genes or DNA sequences to make tiny little changes in the genome of an organism. Now let's look at some tools of biotechnology. First is recombinant DNA technology. In recombinant DNA technology, we combine small DNA fragments from different organisms to create a genetically modified organism. It involves cutting DNA using restriction enzymes and then joining those fragments using DNA ligase. Then a vector such as plasmid or a virus are used to transfer that recombinant DNA into the host cell. Now, the application of these techniques are that we are able to synthesize insulin, protein, and vaccines and other genetically modified crops. And the key importance of this technique lies in the discovery of restriction enzymes for which the scientists also received a Nobel Prize, and the availability of other important tools such as DNA ligase, cloning vectors, and the host organisms. So we are able to synthesize insulin in bacteria, but that insulin is used in human beings who lack the production of enough insulin, for example, the diabetic patients. Another important tool of biotechnology which has been recently discovered is CRISPR-Cas9 gene editing technology. It is a revolutionary tool which can precisely edit DNA of an organism. It utilizes a small RNA that can target specific DNA sequence. Then it allows for insertion and deletion or even alteration of genes in the genome. This technology has been used for treating genetic disorders, for developing disease-resistant crops, and for also performing functional genomics. The advancement of this technique, or the advantages of this technique, lies in its high precision, its cost effectiveness, and also the ease of its use. Another very important tool in the field of biotechnology is the PCR, or the polymerase chain reaction. In the polymerase chain reaction, we are able to amplify one DNA sequence and make multiple copies of that DNA sequence. So you are able to use one small primer, and based on the sequence of that primer, we can amplify the sequence of DNA by using a reaction called the polymerase chain reaction. We don't need to go into the details of that technique, but it's important to know that polymerase chain reaction is used for amplifying DNA sequences. So what is the application of this technique? This technique has been extensively used in the diagnosis. For example, if a person is infected by a virus, you are able to find that the virus is present in the body of a patient by amplifying one gene of that virus. PCR technology is also extensively used in forensic investigations. For example, when there is a crime scene and the criminal cannot be found, you can look for the traces of their DNA at the crime scene by amplifying that DNA and then try to identify who could have done that crime. This technique is also used in genetics research or even parental testing when you want to find who are the actual parents of a person. The variations of polymerase chain reaction are qPCR, which is the quantitative form of PCR, and RT-PCR, which is reverse transcription based PCR. Gel electrophoresis is used for separating nucleic acids or proteins based on their size and charge. DNA, RNA, proteins move through an agarose polyacrylamide gel under the electric current. The use of this technology is for analyzing PCR products, DNA fingerprinting, and for purity analysis for the samples obtained from DNA or proteins. There are several variations of this methodology by using different kinds of stains, and the goal is to be able to better resolve or visualize the samples in the gel. Microbial fermentation is another very important biotechnology tool which utilizes microorganisms to convert organic substances into desirable products under anaerobic or controlled conditions. This technology has been used for the production of antibiotics, enzymes, biofuels, and fermented food, for example, yogurt or beer. Industrial fermentation is performed at a very large scale, and there are several variations of this technique available. Let's look at some common applications of cloning technique now. In the field of medicine, cloning is used for stem cell therapy, where it has been applied for treatment of diseases like Parkinson's disease or diabetes. Cloning of a gene in some bacteria or lower organism is used for production of insulin or other growth hormones, and cloning can also be used for correcting some genetic disorders in an organism. In agriculture, cloning has been used for improving meat and milk production and for creating pest-resistant or high-yield crops. And you must have come across the term GMOs often in a grocery store, which means genetically modified organisms. So cloning has also been used in the field of research for investigating genetic diseases and genetically identical models for reducing the variability in the results. Applications of biotechnology includes creation of vaccines such as mRNA vaccine for COVID-19 or creation of monoclonal antibodies, which also provide you some level of immunity and have been able to combat several diseases. PCR is a tool of biotechnology which is used for testing pathogens, such as qPCR or RT-PCR was extensively used during the COVID-19 pandemic for screening people if they had disease or not. Environmental biotechnology includes bioremediation, which is the use of microbes for cleaning oil spill or removal of heavy metals, or biofuel production, which is producing energy from algae or other organisms instead of fossil fuels. In industry, biotechnology has been used for synthesis of detergents and food processing, and for synthetic biology, which is building custom organisms for performing different tasks. Now let's look at the case study of cloning of Dolly sheep. Dolly sheep was the first mammal that was cloned from an adult somatic cell. So a somatic cell from the mammary gland of a sheep was obtained, and the DNA was put into the unnucleated egg cell, and the whole organism was created with this technology called the somatic cell nuclear transfer. This event was very significant because it proved that the specialized adult cells can be reprogrammed to generate the whole organism. For example, the cells were only taken from a mammary gland, which is the milk production gland, but the milk producing gland cells could actually create the whole organism. So this gave us some hope for regenerative medicine, where we could use the adult or mature cells and reprogram them to create other kind of cells. For example, if somebody has defected blood cells, we could reprogram the cells to create healthy blood cells. The dolly fish went up to live for 6.5 years, after which it was euthanized because of progressive lung disease and arthritis. It did raise ethical debate regarding whether there should be cloning of animals or human beings allowed or not. Because if we allow the use of this technology, then we are defying nature and we are actually manipulating the whole genetic makeup of a population to make them better or superior than the others. Now let's see how cloning can be used for saving endangered species. Przewalski horse was the last surviving species of wild horse, which had only around 2,000 individuals remaining. Scientists in 2020 were able to clone Przewalski horse from the DNA preserved for over 40 years, and the foal was named Kurt and was born in San Diego Zoo. Again, the methodology used here was somatic cell nuclear transfer, similar to the dolly fish. For this purpose, a domestic horse was used as a surrogate mother. The significance of this technique is that it increases the genetic diversity by introducing lost genes back into the gene pool and by preserving and restoring endangered species. But again, it raises ethical concerns that is it okay to de-extinct the species which have been lost because of a very natural process? Now let's look at some ethical concerns regarding these techniques. Human cloning, for example, if we are able to create identical individuals, so there is a chance that a criminal is able to create his or her clone and then they can escape the judicial system. And the individuality of human beings is lost because you can have multiple versions of the same human being, so you cannot associate specific characteristics with one person. Animal welfare is concerned with suffering of cloned animals during the experimental process. So it can be a harsh treatment or unsuitable conditions, and it will put animals through a lot of distress when they undergo these experimental procedures. The biodiversity risk is related to the impact that these genetic homogeneity will have on the ecosystem, because we'll end up producing same kind of organisms and we will lose the genetic diversity. Regulatory issues are concerned about balancing innovation with its social impact. Think about having multiple versions of yourself. How will you be able to associate with your society or have your identity in the society or have even emotional connections with people around you? But like any other situation, cloning has its pros and cons. It can have several applications which can be advantageous for our society. For example, advancement in the field of CRISPR-based cloning has helped us deal with diseases, especially genetic diseases. And it has also helped in creating personalized medicine when we are able to clone patient cell and then test drugs and find out which drugs are actually effective, and then you don't have to give all those drugs to the patient because the drugs can have side effects. By using cloning methods, we can synthesize synthetic organisms which can be used for industrial purposes like synthesis of biofuel or fermentation processes. There are several organizations which are working on global policies for use of regulatory biotechnology tools through which we can regulate how much biotechnology should be used and what should be the limit of its applications. So to summarize, cloning and biotechnology hold transformative potential across fields like healthcare, agriculture, and even conservation. Applications range from treatment of genetic diseases to developing sustainable solutions. But ethical consideration and regulations are crucial for the advancement of this field.